Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaute Envold. In present day neuroscience, it has almost become a dogma that memories are stored in synapses. But that hasn't always been the case. In the 1950s and 60s, a popular idea was that memories are stored in molecules inside neurons, rather than in connections between them. There were good reasons for that. It was, for example, observed that flatworms, small worms comprised of some tens of thousands of cells, retained some memories after their nervous system had been removed, and that one could even transfer memories from an animal by eating their brain. This implied that the memories had to be stored in robust molecules. However, the idea of a molecular memory fell out of favor in the 1970s, partially due to the discovery of long term potentiation, LTP, of synapses in the early 70s, and partially because some questionable and sometimes bizarre experiments with rodents eating each other's brain gave the molecular memory idea a bad reputation. And during the last decades, the synaptic theory of memory has almost become a dogma. At least I never questioned it. Now the situation may change, however. A few weeks ago, I came across the paper, The Molecular Memory Code and Synaptic Plasticity, a Synthesis, written by Sam Gershman. I liked it since it soberly addressed apparent limitations with the synapse-only theory of memory, and also suggested how synaptic and molecular memory may function together. As the question of the biological foundation of memory obviously is a key question for the field, I invited him to the podcast. Sam Gershman is a professor of psychology on Harvard, where he leads the computational cognitive neuroscience lab. In the podcast, we talk about various problems with the synapse theory of memory. For example, the relatively short time scales involved in experiments on spike timing dependent synaptic plasticity compared to the often long time scales over which we can remember things. Many of us can, for example, remember our first day at school, even when it happened decades ago. Can information stored in synapses really last that long? We also talk about candidates for memory molecules like RNA and proteins, and also possible ways to store memories inside the nucleus of cells. One possibility here is a methylation of DNA, one of the mechanisms known to store so-called epigenetic information. Next, Sam talks about his idea for how the combination of molecular memory and synaptic plasticity may work, before we round off with a discussion of sociological challenges of introducing new ideas, questioning established scientific dogmas in the field. Are we sometimes dismissing ideas for the wrong reasons? As for all episode information about the possible episode and relevant links can be found at the webpage theoreticalneuroscience.no. If you want to support the podcast, go to patreon.com/theoreticalneurosciencepodcast and choose a membership of your liking, for example at the cost of $1 per month. As a member, you will get access to a video version of the podcast recordings, as well as episode transcripts. And note that there is a 7-day free trial period when you sign up. I also have a YouTube channel called Theoretical Neuroscience Podcast where you can watch the videos as well. I will be most grateful if you become a Patreon supporter to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help spread the word about the podcast to other people in the community, on social media or otherwise. Okay, here is Sam. Yeah, so welcome to the podcast, Sam Gershman.
Hi, thanks for having me.
Yeah, so the topic of today is sort of the memory and sort of the biology, the cellular biology of memory. But before going there, I thought I should sort of, that you could tell us a little bit about your lab at you're at Harvard, right, in the psychology department.
Yeah, so I'm a professor in the Department of Psychology, and I'm also affiliated with the Center for Brain Science, and more recently, a newly formed Kebner Institute for the study of biological and artificial intelligence. And my lab studies computational cognitive neuroscience, broadly construed, and that ranges from experimental studies in humans and non-human animals, all the way down to single-celled organisms, which we'll probably talk about. And our experimental research is tightly integrated with our theoretical research. So we develop computational models
Mhm.
and try to test them experimentally.
So computational models at what levels? Like, I mean, you like a Bayesian kind of approach, or is that sort of like typical way to go about it,
So,
or?
well, our models are implemented at a number of different levels. Some of them are more focused at the level of cognition. Um, and then some are more focused at the level of neural mechanisms. And with respect to the stuff that we're doing now in molecular biology, we're going to even lower levels of detail towards the underlying molecular biophysics.
Mm. Okay, cool. So now, I mean, the reason I sort of asked you to join on this podcast sort of was I came across your paper, which I think is out on Archive. Maybe it's published somewhere also. It's called a molecular memory code and synaptic plasticity, a synthesis. So there you sort of raised this question or question this almost dogma that has I mean, that sort of I grew up with and I'm still affected by. I would say is that the memory in cells lies in synapses. So you sort of argue that it well that it might be sort of have like this molecular component as well. But first, before we go there, sort of what is what is how do you define a memory?
I would define a memory as any record of the past that is available in the present. And when I say available, I mean available for computation by some
Mm.
system.
Yeah, and in the paper you talk about the structure of memory and distinguish that from the content of memory. So what is the structure of memory?
The structure of memory is basically the kind of coding language in which information gets coded, right? And
Mhm.
that could be at the level of synapses or molecules or something else.
Mm. But and in some sense is independent of what is really stored in the memory.
Yeah, that's right. I mean, and that kind of distinction is familiar to anyone who's
Mhm.
worked with computer memory, right? Because we have mechanisms for storing information digitally that are independent of the content.
Mhm. And like the textbook story of this, if you talk about the structure of memory, is that it has to do with like synaptic strengths and associations set
Mm-hmm.
up by that, right?
Yeah, that would be the standard story.
Yeah, and the standard textbook story of the content is sort of like association between things, or
Yeah, exactly.
yeah. So anyway, I mean, the candidate theories for memory. I mean, the established or the most established idea in the field is that it's synapse only, right? That's what we learn about in the textbook. And and where did it come from? I guess what's the dominant reason we believe that or why that has been established? I guess is LTP, long term potentiation. Is that the key element of that?
I would say there's a few different strands of thought that have converged on this idea and kind of mutually support each other. Um, one is a set of old ideas about the nature of memory that came out of um, empiricist philosophers, but really in some ways go back even farther to the ancient Greeks like Aristotle, um, and um, those kinds of empiricist conceptualizations of memory were framed in terms of associations between images or ideas of some sort. Um, and there were a few things that happened in the 20th century that really reinforced that picture. One was the discovery of associative plasticity mechanisms like long-term potentiation.
Mm.
Um, and then the other was the development of formal associative learning models like the Roscoe-Wagner model, which were
What did you say?
really
That
successful.
what
The
model?
Roscoe-Wagner model of
Okay.
Pavlovian conditioning
Mm.
and lots of descendants of that, which
Mhm.
um, recapitulate some of its central ideas, those were extremely successful in capturing many aspects of animal learning. Although today they're still controversial.
Mm,
But those two pieces of thinking, one about the biology and the other about the psychology, mutually reinforced each other.
mm. So this Hebbian learning, I guess this idea that was, I think it was, well, at least it's most famously posted in this book by Donald Hebb in 49 or something. So that
Mhm.
is that part of this Pavlovian tradition, or is that part of this?
Um, yeah, I think that that was certainly part of it. I'm not sure that Hebb wanted to tie himself too strongly to Pavlovian conditioning, but Pavlovian conditioning is still considered the kind of gold standard of associative learning phenomena. Um, yeah, and I think that Hebb was thoroughly in that tradition.
Mm. And okay, so then we're at LTP, long-term potentiation, which was then seen in, I guess it was discovered here in Oslo with Bliss and Lermo, and
Mm-hmm.
but that was sort of that is sort of the strengthening of synapses related to like stimulation of certain like electrical stimulation, right?
Yeah, that's right.
Yeah. So what is the key elements of that
Sorry, what do you mean the key element of which
of
thing? Of
LTP? So
Yeah.
okay, so what's the main
Yeah.
observation with LTP?
Well, the main observation is that the coordinated activity of presynaptic, postsynaptic neurons tends to strengthen their connection, in the sense that if the presynaptic neuron becomes active, then the postsynaptic neuron becomes more likely to be active following
Mm.
potentiation.
Huh. So and so that is sort of like a, I guess, like a well established fact that we have this LTP and LTD that is sort of initiated or set up by certain kind of stimulation protocols. But what is the connection between, or what is the, how do you know that, how have people established that this is somehow related to memory?
Yeah, that's a great question. So probably the place where this is most well worked out was in the amygdala, largely thanks to the efforts of Joe Ledoux in his lab. Um, and there, the idea was that neurons in the basolateral amygdala um, are signaling this learned association between, in that case, between a conditioned stimulus like a tone or a light, and an unconditioned stimulus like a shock. So this was most extensively studied in Pavlovian fear conditioning. We know that there are neurons in the basolateral amygdala which fire in response to the conditioned stimulus, and that firing pattern emerges over the course of Pavlovian conditioning. If you lesion the amygdala, you um, you eliminate this associative process.
Mhm.
And the question was whether there are synapses at these neurons that are encoding that association. And they did a lot of work to show that one of the things that was happening there was that during conditioning, there was trafficking of AMP receptors to the postsynaptic membrane. And they even showed that during extinction, so if you just present the conditioned stimulus by itself a number of times, um, the animal will stop freezing, and that's accompanied by removal of AMPA receptors from the postsynaptic membrane. So that was one of the findings that led to uh, that led to this idea that trafficking of receptors to the postsynaptic membrane was an important
Mm.
correlate of this synaptic strengthening, and the causal link um, to learned behavior was established in a number of different ways. For example, by uh, administering protein synthesis inhibitors to the amygdala, which prevented the synthesis of plasticity-related proteins, which were necessary for the production of those receptors.
Mm.
And that could impair memory if it was done shortly after the conditioning episode. Um, and they also had various kinds of pharmacological manipulations of AMP receptors. And then later on in the 21st century, with the development of optogenetics, people started using those tools to optically manipulate memories. So there were a whole cluster of findings that all kind of pointed in this particular direction, that there were synaptically stored memories, and that there was a particular molecular mechanism underlying that.
And also, I think, I mean, this Morris water maze is also, when I asked about this to some colleagues, they
Mm-hmm.
mentioned this Morris water maze. It's also sort of like a strong indication of the, well, proof to the point that there are sort of like, do you think you need NMDA receptors or something to form these memories?
Yeah, that's right. That's right. So you need NMDA receptors to produce long-lasting, long-term potentiation, and disrupting those NMDA receptors will disrupt learned performance in tasks like the Morris water maze.
And but you, I think you point to what you say is a logical flaw. That, I mean, it's not necessarily so that, I mean, that it could be still be that the things are stored in molecules, but that you need
Yeah.
synapses to just to implement the memory, sort of.
Well, I would put it a little bit differently. The idea is that you need synapses to express memory, right? If you don't
Mhm.
have any synapses, there's no way for a memory that might be stored in a molecular format, hypothetically. There's no way for that memory to get to behavior, right?
Mhm.
So if you mess up synapses, you obviously will mess up learned performance, but that doesn't mean that the synapses are themselves the terminal storage site of the memory.
Mm. But I guess you could also imagine, couldn't you, that actually even without synapses, there could be some diffusion of molecules between cells, and that could also affect behavior, but maybe then on a much slower time scale. So that's
Yeah, absolutely. And in fact, there's evidence that there is something like that happening in the brain.
okay.
We know that there's lots of exchange of molecules between cells. The actual behavioral and cognitive implications of that have yet to be worked out.
Mm.
Um, but
Mm.
that is one possibility.
And okay, so now we talked about essentially like the synapse only ID of memory, and then you have the sort of molecules only ID. Maybe I guess with Randy Gallistel, it sort of has
Mm-hmm.
been his at least a well-known proponent for this ID. So what is the idea here? That it's that memory is somehow stored in? Is it only in the soma, or is it everywhere in the cell? Or
Um, there are different possibilities, and there's not really strong evidence for any one particular possibility. So Galstel's arguments are mostly based on kind of in principle arguments. One of the issues that Galstel points out is that um, it's energetically very costly to maintain memories synaptically. To achieve a particular bit rate, essentially, you need to use orders of magnitude um, more metabolic resources compared to if you stored it, for example, in a polynucleotide sequence like RNA.
Mhm.
Um, Now, we don't know whether memories are actually stored in polynucleotide sequences, although there's some suggestive evidence that RNA, particularly non-coding RNA, is involved in regulating memory in some way that's separate from just the synthesis of plasticity-related proteins. Um, so Gauss's arguments are based partly on sort of these arguments about the energy efficiency and thermodynamic stability of memories stored in polynucleotide sequences. I mean, we have one obvious existence proof that polynucleotide sequences are suitable for storing memory, which is DNA.
Absolutely.
Now, we don't think that most memories are stored in DNA, but it's an example of a kind of stable and efficient memory code that's stable enough that it can permit propagation of genetic memory over many generations.
Mm.
Um, and then Gaustel So arguments are also partly based on a critique of the connection between um, synaptic plasticity and learned behavior. And he basically has a number of arguments that the properties of behavior in terms of the timing of the behavior, what is learned, how quickly it is learned, um, don't quite line up with properties of synaptic plasticity. And that's something that's in some ways acknowledged by the people who study synaptic plasticity, where they appeal to other kinds of mechanisms that seem to lie outside of the synapse. And for that, Galstel harshly criticizes them because he says that they're basically appealing to things that are outside of the core hypothesis.
I mean, we're coming back to these arguments later in the podcast, but but I guess I mean you actually have in your theory, which also coming back to that is actually has a role for both the synaptic and the distinct roles for synaptic memory and sort of molecular memory. But I mean, sort of like if you look at the history of models for memory, I guess until until the 60s or maybe early 70s when LTP came along. And I don't know when these like experiments by Joe Ledoux, was that also 70s, 80s or
No, that was later. That was more like 90s,
yeah.
2000s.
I
Yeah.
guess also this Maurice Watermaze was also 80s or I don't
Yeah.
know. Yeah, something like 80s, 90s. But anyway, until the 70s, the main idea was or at least one of the ideas, at least I was experimentally studied, was this molecular memory. And I think you've written this, you wrote this nice paper about single cell memory, where you pointed to this interesting person, Beatrice Gelber. What was she
Yeah.
doing?
Yeah. Beatrice Gelber was an interesting character. In the 1940s, she was a housewife with grown children, and something inspired her to go back to grad school. And she ostensibly went to grad school to study psychophysiology, which she did. But while she was doing that, basically completely on her own, she started studying paramecia, which is a single-celled organism, a kind of ciliate. So it's covered with cilia that allow it to swim around. And it swims around in pursuit mostly of bacteria that it consumes. And part of the reason that she was studying it was that there was a history that had predated her, going back all the way into the 19th century, where people had been fascinated by the seemingly complex behavior of this organism and had tried to characterize it in various ways. The most famous work on this topic was by Herbert Spencer Jennings. He wrote a book called The Behavior of the Lower Organisms that was published in 1906.
Oh.
But there wasn't really that much done systematically on studying learning in these organisms, and certainly nothing very parametric and comprehensive. And Gelber was coming at a time where, at that point, people already knew quite a lot about the parametric properties of classical conditioning. And so essentially what she started doing was developing a classical conditioning paradigm where she could study these things. And so she undertook what remains the most systematic research program on classical conditioning in paramecia. And essentially what she did was she would swab platinum wire with bacteria. Now, this platinum wire did not have any attraction on its own for the paramecia. So if you just put the platinum wire in by itself, this paramecia wouldn't do anything, unless, of course, you tap them with it and they'll swim away from it. But if you covered it in bacteria, the paramecia would swim and kind of attach themselves to the wire or swim around its vicinity.
Because they eat bacteria, or
Yeah, because they eat bacteria. Now, what she then did was she would pair this wire with bacteria several times, and then after doing that, she would put the wire back in with no bacteria on it. And what she showed was that the paramecia would still swim and attach themselves to the wire even in the absence of bacteria. So she argued that the paramecia had acquired a conditioned response
Yeah.
to the wire. And she
So
did a bunch of controls to try to rule out alternative explanations. And she also did a bunch of parametric studies, like looking at how long the memory is retained. It turns out that it can be retained up to 24 hours, which is quite long in the lifetime of a paramecium. And she did things like show that you could produce spacing effects, like if you space out the trials, learning is faster. So it was really quite an impressive set of studies. But at the time that they were published, they were attacked quite a bit by skeptics. And I think one of the problems that Gelber had with this whole setup is that it's quite hard to rule out the possibility that there's some kind of residual concentration of bacteria that's driving this effect. Plus, there's no known mechanism by which cells could sort of sense and navigate towards this wire in the absence of the bacteria. So there are a number of problems, but I think she was really unfairly forgotten, basically criticized and then forgotten. And it was only recently, but partly actually due to our own research into her work, that her reputation was revived. And now at least there's a Wikipedia page about her.
Mhm. And then, I mean, this is a single cell, single cell organism,
Mm-hmm.
but they also, a bit later, right, in the 60s or maybe earlier, you also write about these flatworm experiments,
Mm-hmm.
right? Where,
Yeah.
and this is I just looked it up, it has about 25,000 cells. So this is sort of a much more complicated organisms. And
Yeah.
so what were sort of well, what was sort of the findings they had with the flatworms, that it had some interesting properties?
Yeah. So these were planarian flatworms. They
Mhm.
were mostly studied starting at the end of the 19th century for their regenerative properties. So they have this remarkable capacity to regenerate from tiny fragments, like less than 1% of their body mass. Um, and in the 1950s, it was noted that the cells can also do classical conditioning. So you can, for example, expose them to a light that's paired with a shock, and then they'll emit a conditioned response to the light over multiple pairings. That basically they'll contract in response to the light. So putting two and two together, the researchers wondered, okay, if I cut off this worm's head, it will grow a new head. Now, if I had trained it before cutting its head off, it will get a new head. Will the new head remember what the old head had learned?
Mhm.
Um, and the answer, remarkably, was yes, that it will exhibit some memory of of its conditioning experience after losing its entire brain. Okay? Um, and that result has been replicated more recently by Mike Levin's lab.
Yeah.
Then they started taking it farther, and this is where we get into some murky territory where it's a little bit unclear what's fact and what's fiction. And actually, one of the things that I'm doing right now in collaboration with Mike Levin's lab is to try to replicate some of these studies that I'm about to tell you about.
Mhm.
So they knew that the planaria would cannibalize each other under some circumstances. So what they did is they trained one worm, and then they ground it up and fed it to an untrained worm.
Mhm.
And their reasoning here was that if the memory was stored in some kind of molecular format, particularly a format that could survive decapitation, then maybe you could actually transfer that molecular information from one organism to another. Um, and what they found was really astonishing, which is that the untrained worm, after ingesting its trained friend,
Mhm.
produced a conditioned response.
Yeah.
Okay. And so then they wanted to take it even further and ask, okay, can we narrow down what the candidate molecules are? And at that time, molecular biology was really in its infancy. So we're talking about the late 50s, early 60s. Um, and they knew about DNA. Right? They knew about RNA, although not all the different kinds of RNA. So they mostly knew about messenger RNA. They thought that maybe RNA could potentially encode information. And so what they did is they extracted RNA from the trained worm and injected it into the untrained worm and showed that that could also produce a transfer effect. And that was actually that's the study that I want to reproduce, because that was probably one of the best studies that had been done on this topic. That it was high powered with good experimental controls, and those results seem to have been produced several times in the 1960s. After that, people got really excited, and everyone was trying to transfer all sorts of different things with all sorts of different species. And there, the results became much less reliable. And one count has it that about half the studies worked and half of them didn't work. And so
This
eventually,
was still on this flatworm.
no, on all sorts of organisms
Okay.
like rats, mice, fish,
Yeah. Huh?
chickens,
Yeah.
hamsters. So people were doing all sorts of things, but there
Yeah.
wasn't really a well standardized procedure. They didn't understand the molecular basis. They never figured it out. They didn't understand why it worked and why it didn't work sometimes. And so eventually the whole area of research lost steam. And what ended up happening after that was that it was sort of internalized into neuroscience that the whole program of research was kind of a false start.
Yeah.
And so today, if you read anything about this, or many students don't know that this ever even happened, and the articles that are written about it today basically frame it as a curious but sort of embarrassing episode in
Yeah.
the history of neuroscience.
Exactly. Yeah.
But what's yeah.
That's
I mean,
how
I'll
I
just
effort to continue.
Oh yeah, I was just gonna say. I mean, what's interesting is that now it's gradually creeping back, because a number of research groups have shown that you can actually transfer transfer memory via RNA in several different organisms. Um, uh, David Glansman has looked at this in aplysia, um, and Colleen Murphy has looked at this in another kind of worm, C. elegans. So it may be coming back. And there's a bunch of other suggestive findings, not specifically about transfer, but in this vein that suggests that memory might be encoded in a molecular format.
Yeah, because I think because I heard just this when I read about. I read well back in the days. I read about these rodent experiments, and that sounded sort of me like a bit crazy and bizarre, and it sort of it was, yeah. So this was clearly something that was it's more of a story of how stupid people have been in the past, rather. So it's sort of an interesting sociological thing, right? That we have been through this almost like molecular memory winter for 50, 60 years,
Mm-hmm.
just like they
Yeah.
go through, and now it's coming back, and we have like these ideas which are dismissed for the wrong reasons. Maybe if it gets some bad rap, and then this new candidate, of course, this new thing with synaptic plasticity coming in, and so it's interesting how vulnerable we are, maybe as a scientific field, to following fads and dismissing things for the wrong reasons.
I mean, I don't think it's quite that bad. I think that the people who study memory from a synaptic plasticity viewpoint have very good reasons to believe what they believe, right? There's just been so much more work on it than there has been on um, these other things that we were talking about.
Yeah.
And so to overcome that would require just quite a bit of work. And nobody wants to get over that energy barrier because it's deeply unpopular. So it requires actually, you're talking about the history of human stupidity. In some sense, to get over the energy barrier requires a bunch of stupid people, people stupid enough to undertake deeply unpopular research, right? People like me. So
Yeah. Yeah. No, because I heard this interesting podcast with Glansman, right, on the Brain Inspired podcast. So but he also mentioned that, I mean, one thing is that it sort of falls out of fashion, but it's, I mean, he's quite well established with his synaptic work. And then when he tries to explore this molecular memory thing, he has much harder time getting funding. So it's
Yeah.
also this practical thing, right? That if you're doing something a little bit unconventional, it's difficult, and it's more difficult to get funding, maybe.
Yeah, I don't know. I think yeah, there's all sorts of sociological issues here. I mean, one thing is, you have to find a way in that doesn't alienate people, right? Um, so I think there is some history here, particularly by Gallistel, of basically wagging your finger at people and telling them that they're wrong,
Yeah.
and people don't usually take well to that.
Mm. Mm.
So I actually I think it's too black and white to say that the people studying synaptic plasticity are totally wrong, and so that's
Mm.
actually where I kind of part ways with Gallistel, because I actually think that synaptic plasticity and memories stored at synapses are actually a really important part of the whole picture, but they're
Yeah.
not the only part of the picture, right? And I
Mm.
think that there's a way in where we can all kind of have what we want. We can address the inadequacies of a pure synaptic memory account, while not completely abandoning it.
Mm. So now to the, like, limitation or problems with this synapse view,
Mm-hmm.
and one is the, which I think you alluded to already with that Gallisel mentioned, is about time scales, because at least this spike timing, the dependent plasticity rules, they are typically, well, it's more on the millisecond or second time scale maybe, and this, like LTP and these things are also, I mean, typically on hours or something. But we
Yeah.
remember our first day at school, right? So I guess the large timescales of these associations or these memories is a challenge for the synaptic viewpoint.
Yeah. Well, there's a few different things happening here. So one is about um, the time window of uh, proximity between events, such that you can associate them with each other. Right? So,
Mhm.
so when we say that one event was paired with another, we're making a claim about temporal proximity. We said that uh, when the tone and the shock appear close enough in time to each other, then they will be associated with each other, right?
Mhm.
Um, and that raises the question, how close is close, right? Um, uh, and there's an analogous question for the synaptic plasticity account, right? So what spike timing dependent plasticity shows is that there is a critical time window in which the presynaptic neuron has to fire before the postsynaptic neuron, and it's on the order of 50 to 100 milliseconds, so very short. Um, and if it doesn't fire within that window, you won't get any long-term potentiation. And in fact, if the postsynaptic neuron fires before the presynaptic neuron, you'll actually get long-term depression. Um, with Pavlovian conditioning, there's actually a huge range of timescales in which you can see conditioning effects depending on the preparation. And what I mean by this is that you take the conditioned stimulus and the unconditioned stimulus, you vary the delay interval between the two, and you usually see this inverted U shape where really short intervals actually are slightly worse than slightly longer intervals, and then it goes down again. But the x-axis of those plots differs quite a bit between preparations like eye blink conditioning versus Pavlovian fear conditioning or something like that. But there's an even more fundamental issue here. Well, so actually, let me first say that first of all, the peaks um, of those curves are much longer than 50 to 100 milliseconds. Okay?
Mhm.
So that's already potentially a problem for an spike timing dependent plasticity account of those phenomena. But the other thing, and this is the thing that Galstel really emphasizes, is that there is no real critical time window, that these plots are actually an illusion. And the reason is that um, there's another thing that we know about Pavlovian conditioning, which is that um, you can get spacing effects, which means that if you space out the trials, so you increase the intertrial interval, conditioning will happen more quickly. Okay, so you have these two time variables: the length of the interstimulus interval, which slows down conditioning if you make it longer, and then the length of the intertrial interval, which speeds up conditioning if you make it longer. And it turns out that there is, at least for a pretty wide range of these intervals, a quantitative law that says that um, what matters for the speed of conditioning is actually the ratio between these two. Okay, and in fact, if you increase the interstimulus interval while holding the intertrial interval fixed, in other words, if you double the interstimulus interval and you also double the intertrial interval, then the speed of conditioning will remain the same, even though now the interstimulus interval is twice as long. Okay, and that seems to hold up pretty well, but it's extremely troublesome for an account of temporal proximity as the driving force behind association formation, because it becomes impossible to state what a critical time window actually is for conditioning. It suggests that there is no critical time window. At least that's how it's been interpreted.
Yeah, because it's I mean you I mean even I mean I think you also mentioned this, that like with X-ray induced sickness, right? It on I guess if you get sick by getting from high doses of X-rays,
Mm-hmm.
then it takes an hour or something
Mm-hmm.
before you get sick, and you still have to make this connection. I mean,
Yeah.
this we talk about, I guess, rodents or something, right?
Yeah, so what you're referring to is conditioned taste aversion. So first
Hmm.
of all, just take regular conditioned taste aversion, where um, you eat something and it's paired with illness, and you get sick a few hours later.
Yeah.
This is mostly done in rodents.
Mm.
The rodents
So
will,
sometimes in humans also.
yeah, I mean, it's certain. Anyone who's ever gotten food poisoning knows
Yeah,
exactly what we're
yeah,
talking
like
about,
yeah, yeah.
right? So you learn an extremely strong aversion, and it only takes one to whatever you're eating, and it only takes one
Oh, shit.
experience like that to induce a strong
Mm,
taste aversion. And
mm,
what's interesting about that phenomenon is that the illness doesn't materialize usually until at least several hours, and sometimes even longer after you've eaten something. Um, and the reason that's interesting is because all sorts of things will have happened in that interval that have closer temporal proximity than the food that you ate, right? So, why is it that you associate the illness with the food, in the sense that you learned an aversion to the
mm,
food, but you didn't learn an aversion to any of the other things that happened in between eating and getting sick, which happened
mm.
closer in time to
Yeah,
the illness, right?
yeah.
And it's um, a really striking demonstration of this issue were these experiments that were done by Garcia and Colling in the 1960s. And this is what you're referring to, where you can you can irradiate an animal and induce gastrointestinal illness several hours later. And they were interested in whether you could get animals to learn a place aversion. So basically, the animal is in a particular location.
Mm,
You apply the X-rays, induce illness several hours later. Will the animal avoid the place where it was exposed to the X-rays?
mm,
And interestingly, you can't do that. You cannot induce a place aversion through gastrointestinal illness.
yeah.
And you also can't induce taste aversion through other kinds of punishment, like electrical shock.
Ah,
Um, and this was a bit um, uh, troublesome also for associative learning accounts, particularly the accounts which saw the laws of associative learning as really general.
mm,
That essentially the brain, the most extreme versions of these ideas, the brain is sort of a blank slate, and it gets these experiences. It doesn't really matter what they are. Anything that has a systematic association will be acquired according to these laws. But what these results seem to show is that the laws of conditioning, whatever they might be, are not so general, because they only because some things are more associable with other things than other kinds of pairings. So it suggests some sort of domain specificity in the laws of learning.
So yeah. So okay. So then, if you want to, well, if you're focusing on the neuron level then, and synapses, and so then the question is sort of, well, I guess also spines. I mean, many synapses are, at least, are on spines, and the spines don't last so long either, do they? I mean, they are
Yeah.
also rapid turnover. So
Yeah.
what are the tricks, if you sort of, well, what are the tricks the neurons can do in order to maybe to deal with longer time scales and remember for longer times?
Yeah. So if you're looking at it from a purely synaptic plasticity point of view, this has been an area of quite a bit of research
Yeah.
in the last few years, because people have been aware of the fact that there is a lot of spine turnover, but the implications of that have been somewhat controversial, right? So one read of that is that it's just impossible to store memories synaptically because of that turnover, but that's probably too extreme of a conclusion, because there's a bunch of ways around it, and people have developed computational models that implement these ideas. Um, so one possibility is that maybe memories are stored at just a small fraction of the synapses. So it's a sort of sparse coding kind of idea. And indeed, empirically, there are there usually is a small subset of synapses of spines that seem to resist turnover and last a lot longer.
Ah.
So maybe that's the basis of long-lasting memory.
Mm.
Um, Another possibility is that if you think about memory storage from a population coding point of view, then uh, even though individual synapses are lost, the memory might still be encoded at the population level.
Mhm.
Um, but to make that work, you'd have to either have sort of cooperative reorganization of the code
Mhm.
to sort of repair itself after loss of spines, um, and or some um, updating of the decoder that reads out that memory, right? And this comes into contact with ideas about representational drift in neural systems, which has been studied quite a bit.
Hmm. So I think you also mentioned that this thing, that there could be some a candidate, that there are some kind of bistable switches in, like, some molecules, I guess, inside synapses, that maybe also could have some, like, well, maybe extend some time scales, or do, is that correct?
Yeah, that's right. So there was this idea going back to the 1980s from Francis Crick and then also from John Lisman, that um, hypothesized that bistable molecular switches, could store memory over longer periods of time, because if one of the marks on the switch, like a phosphorylation mark, for example, was disrupted, then via autophosphorylation, it could repair itself. And potentially different molecules could also phosphorylate each other, sort of collectively maintain a memory.
Mhm.
Oh, and John Lisman pointed in particular to CAMK2 as a molecule that had this property, and that stimulated quite a bit of research by his lab and other labs over the ensuing decades,
Mhm.
to try to test whether CAMK2 was responsible for this sort of long-lasting memory. Um, the I mean, a lot has been understood better now about CAMK2. One of the controversies is that um, although CAMK2 is clearly involved in the induction of synaptic plasticity, and that seems to have something to do, at least partly, with the phosphorylation of AMPA and NMDA receptors, um, um, it's not clear that you need CAMK2 for the maintenance of memory. So there were some studies suggesting that if you um, suppress CAMK2, then you can still get memory maintenance, but you don't. Uh, you get memory maintenance even in the absence of CAMK2, suggesting that you don't need it for memory for that function, right? So, but you might still need it for induction of memory.
Mhm. Also, one, I mean, here in Oslo, there's one of my colleagues, Marianne Fyn has studied this what's called perineuronal networks,
Hmm.
so which are some kind of extracellular sugar structure that I think Roger Tsien suggested that they could be sort of like a way to lock the synapses almost, so that
Hmm.
they can't be written on something, or to store them. So I guess
Yeah.
that's so that they somehow, yeah, as a way to freeze memories.
Yeah. I mean, I don't know that much about perineuronal nets. It seems like an interesting idea. And there are a bunch of ideas that go beyond things like AMPA receptors. I mean, the synapse is an incredibly complicated place, and people have speculated about all sorts of structures that facilitate encoding of memory at synapses, like microtubules and all sorts of other things. So it's a little bit hard to parse
Mm,
what the roles of all of these things are.
mm,
And this is one of the tricky things about studying this question, right? Because it's not clear there's any one molecule that's responsible for storing memory, right?
mm.
It could
Yeah.
be a multi-scale cascade of
Mm,
molecules.
yeah. Okay, so now we're segueing into this, like the search for this memory molecule, which you also wrote about in your paper. So, well, first, I mean, I think you mentioned when you talked about this molecular theories for memory that there were sort of like two classes of theories in the 60s for this one, something called the switchboard theory and something called the direct coding theory. What were these things?
Um, Essentially, the switchboard theory was about thinking about memories as kind of like routers of
Mm,
information. And then there's direct coding, which in which memory is directly written onto the molecules.
mm,
And in some ways, what my proposal is about is integrating all of these things or some aspects of these ideas.
yeah. So yeah, exactly. So this direct coding theory, that's more like how we store numbers in computers, right? That's you just
Mm-hmm.
actually put numbers in certain places
Yeah.
and store them. So okay, but what about the candidates? So then you have, okay, so you mentioned DNA and I said, but and RNA maybe in particular for like as a candidate molecule. Is that your favorite candidate?
Well, one of the reasons to think that RNA may be important is because of these memory transfer experiments.
Mm,
But I would say that it's still very unclear, and that's one of the reasons why we're undertaking experimental research to test those ideas.
mm. Yeah, and because I guess you can also this RNA can be packaged in
Yeah.
this little like a little virus kind of thing almost.
Hmm.
So they can sort of actually be transported between cells and also inside cells. So I guess it's all kinds of theoretical possibilities. What do you think of this historical? I mean, if you sort of look back in the if you look historically on this, I mean, like evolutionary speaking, then because back in the day we were only like we're single cell molecules, and I guess the flatworms with 25,000 cells came before us, and so I guess it's and even before life started, it could have be that we were in this RNA world before DNA became sort of established as this permanent storage mechanism. So how strong do you think this argument is that, well, we have, it will be strange to abandon something that we used in the past?
I mean, I wouldn't rely on that too strongly. I think that in general, nature is conservative,
Mm,
um, and a lot of our cellular machinery uh, is the same as much simpler organisms. However, it doesn't follow logically that it has to be the case that, however, those simpler organisms store memory has to be the same as how complex nervous systems store memory.
mm,
So it has to be demonstrated. It's more of a heuristic, a rule of thumb.
mm. And then you have, I mean, I guess in addition to RNA, you have proteins and obviously DNA, and then you have all this methylation process and all these ways to sort of selectively sort of maybe block memories on certain places on these molecules?
Yeah. So in cell biology, one thing that's been extensively studied is the role of marks in the nucleus to store information. So there's marking processes that are experience dependent, and they affect things like the methylation of DNA or the modification of histones in the chromatin um, um, around which DNA is wrapped. And essentially, what these things do is control um, the access of different pieces of DNA to things like transcription factors. So most DNA is not being transcribed at any given time. Its transcription has to be activated, and for it to be activated, that piece of genetic code has to be accessible to um, second messenger systems. And so these marks control access to those systems, and in that way, the cell can acquire uh, sort of longer term memories that control what gets transcribed and ultimately what proteins are produced,
Mm.
among other things. And one of the interesting features of that is that those marks seem to at least partly be passed on to future generations. So you get this kind of epigenetic inheritance of memory.
So how does that work? Because I thought like humans, like gametes or like sex cells or whatever,
Mm-hmm.
they are made when we are a fetus, right? So the thing that is sent off to the next generation, how I just how was I can sort of understand how like a somatic cell in my body could be
Yeah.
affected by these methylations, but how a germs or a gamete could be, that's more difficult to.
Yeah, I mean that we don't totally understand that. You're right that you require some pathway for communication between the soma and the germline.
Mm,
Um, in simple organisms, that's easier, particularly in single-celled organisms.
mm,
Um, Actually, in ciliates, it's interesting because many of them have multiple nuclei. So they have macronuclei and micronuclei, which play the role of the soma and germline. They essentially have a soma germline separation
okay.
inside of a single cell.
Ah,
But there is communication between them.
mm,
Um,
mm. Yeah. But anyway, regardless of whether we can sort of go through between generations, epigenetics in itself is a memory mechanism, right? That's a methylation of say DNA. That is a well, like at least partially. That is some kind of memory if that is sort of history dependent and experience dependent. Wouldn't you say?
Wait, sorry. Which part of it is history
No,
dependent?
I think it's
Yeah,
like epigenetics, right? And
yeah.
if you sort of are, and if it's like experience dependent epigenetics, so it affects like how my cells are transcribed now, or the DNA is transcribed. That is
Hmm.
already some kind of memory, at least in body
Yeah,
memory.
that's right. That's right.
Yeah, so it's some kind of it already is some kind of yeah. The body remember the body remembers. Yeah, as we yeah. Anyway,
Hmm.
so now and then you have this final thing just to be try to be complete. You also have these histones. What histones? That's another level of organization, right?
Hmm. Yeah. So that's another kind of epigenetic mark
Mm.
that controls access of different parts of
So,
DNA to
mm.
transcription factors.
So now there are many sort of like I guess biological candidates for molecular memory, but now I thought we could talk about sort of your idea for how the combination of molecular memory and synaptic plasticity may work. And I think you presented your ID. At Mars three levels, right? As like first the computational problem, the algorithmic solution, and then the physical implementation. So maybe we could start with the computational problem.
Yeah. So the computational problem um, is framed not specifically in terms of memory, but rather in terms of learning and inference. So the problem facing any cell or organism is it's getting signals from its environment. It needs to respond to them appropriately. So it needs to figure out what it should do right now. There's a fast timescale response. And then there's also a slower timescale learning where it's learning about the structure of its environment. And we can frame those things in terms of a generative model where there's some latent cause in the environment.
Okay, so here, okay, so
Yeah.
this is like a Bayesian. So what is okay? First, what is we can just be clear that we have all listeners on board. So what is a generative model in this
Yeah.
case?
So well, I'm trying to explain that now. So
I was sorry about yeah.
there's a let's suppose, right. So let's suppose that there are hidden or latent causes in the environment that are causally producing signals that are measured by a living system like a cell.
So a latent cause, is that something like what is a latent cause? How would you define a latent cause?
So if you get if your mechanoreceptors start activating, right? That's because there was something outside of you that collided with you, right?
Exactly. Yeah.
And you don't get to directly observe that thing
No.
outside. You only get to see it through the prism of your receptors. Okay.
Exactly. So that's a possible cause.
Yeah. So let me make this a bit concrete. We can take an example of another kind of single-celled organism called stentor. It's another ciliate, which my lab has been studying recently. And stentor, unlike paramecium, it can swim, but it doesn't swim constantly. It usually will anchor to the bottom of a pond and filter feed. But if it gets disturbed, it will contract. It's a defensive response. If it gets disturbed enough, it will remove its anchor and swim away. Now, the basic problem facing stentor is that it's in this pond, which has some varying degree of turbulence, because different stentor can be born into different ponds. They don't have a genetically encoded knowledge about turbulence in the pond. And they also don't know about the risk of predation in that particular pond. So that's something that they have to learn. So there's a trade-off, because if it contracts too much, then it will lose the opportunity to feed. But on the other hand, if it doesn't contract enough, then it might fail to avoid a predator. So or something else that could damage it. So the idea is that the inference problem for stentor is what is happening right now, in the sense of, like, am I being attacked or physically threatened in some way right now? And if it infers that it is being attacked, then it should contract. Okay. Otherwise, it should not contract. Right? And then there's a learning problem, which is to learn the statistics of its local environment, which says basically, what's the probability of getting attacked. Right. So maybe I'm in a turbulent pond and my mechanoreceptors are getting stimulated a lot. But that's not actually dangerous to me. And so I should basically attenuate my sensitivity to those signals. And that produces a phenomenon called habituation, where the cell gradually stops contracting if it's exposed to mild stimulus repeatedly. So that I interpret that as a kind of learning phenomenon. It's learning something about the structure of its environment. Right. So there are these two problems: one fast one, inference, and one slow one, which is learning.
Yeah.
Does that make sense?
Yeah, that makes sense. Yeah.
Okay. Okay. So I want to argue that this kind of division of labor, it applies not just to single-celled organisms; it applies to neurons. So a neuron is trying to figure out whether to fire or not, and it's getting signals from its neighbors. And so it has a fast timescale problem of figuring out, should I fire right now? And then
Mm-hmm.
also a slower timescale problem, which is the problem of learning about the statistics of its inputs. Okay.
Mm-hmm.
Um, now, one problem that is often raised for molecular accounts of memory is that it seems hard to have a really fast system for reading out memories.
Mm-hmm.
And what I want to argue is that um, information is actually not read out from the molecular memories directly, but rather um, the information about these like local environmental statistics are being stored in a molecular memory code, but the role of that code is to not directly influence behavior in real time, but rather to influence learning.
Mm-hmm.
What I want to argue is that synaptic memory functions as a kind of parallel memory system, which mediates fast inference.
Okay. Yeah.
And the way in which those two things talk to each other is that the molecularly encoded memory functions as a kind of instructive signal to the synapses to tell it how to adapt the synapses to appropriately reflect the underlying structure of the environment.
Hmm.
And you can actually uh, frame this kind of computation in terms of three-factor learning rules, where you have the two Hebbian components, pre and postsynaptic activity, and then usually often models you'll see these the third factors, which are usually global factors that come from elsewhere,
Mm-hmm.
like neuromodulators, for example, dopamine. But here, what I'm arguing is that the third factor actually comes from within the cell.
Hmm.
Um, so that's where synapses. I want to say that synapses are a fundamentally different kind of memory in the sense that it's a kind of computational memory.
Mm-hmm.
What that tells the neural circuit how to quickly respond correctly um, to its inputs.
Mm-hmm. Yeah. Exactly. So that's a computational memory, and then you say that the molecular memory, I think you call it representational memory,
Yeah,
right? So that's where, I mean, here you say that maybe this is sort of like where they learn how to adapt or how to sort of to, as I was like, learn the facts about the environment, maybe, or something
yeah.
like that. Yeah.
That's right. Right.
Mm,
Because the facts that you're learning are really the parameters of the generative model producing the
mm,
data that you observe. Right. So this is what I mean by the local environment statistics. Like for a stentor, it's something like, what's the probability of getting attacked in my,
mm,
in my local environment?
mm-hmm. Yeah. Huh. Cool. So and then I think you mentioned, in, and then in your sort of theory, which is fleshed out in this well, at least in more detail in this chapter, you say that it's actually there are two things. You can formulate this as an optimization problem where two things are being optimized at the same time.
Mm-hmm.
That both these two aspects of both the representational representational memory and well, the representational part and the computational part. Yeah, cool.
Yeah. So
Hmm.
so um, there's a way to formalize these two um, computational problems, inference and learning, in a unified way, and this is via the free energy principle. But really, I mean, that terminology has a lot of baggage, and so we can
Yeah.
maybe think of it in a more neutral way as essentially a
And
form of
actually, I read
yeah,
your little bit explanation of this free energy principle, the
yeah,
paper, which
yeah,
actually clarified things for me and sort of put it into this context. That is some kind of a specialized case of like the Bayesian approach, right? I mean, it's
yeah.
an approximate way to do Bayesian
Yeah, I
inference.
mean, well, I would say that in some ways, people working on the free energy principle have gone beyond that. But the
Okay.
way I'm using this is essentially a way to do approximate Bayesian inference.
Yeah. Yeah. Mm.
And this, well, we don't have to go into the technical details, but essentially, this utilizes ideas from variational approximations that were developed originally in physics and then made their way into machine learning. And perhaps the most important idea that was developed in machine learning over the last few decades was this idea of amortized variational inference. And the idea there is that um, you have some kind of persistent memory of um, uh, persistent computational memory that allows you to learn how to approximate not just one probability distribution, but a whole set of probability distributions where the different
Okay.
probability distributions are distinguished by the sensory inputs, right? So every time you get a sensory input, you have to make an inference about the latent variables, the latent causes, conditional on those sensory inputs. And then when you get new sensory inputs, you have a new probability distribution.
Okay.
But there's a lot of shared structure across these probability distributions, and amortized approximations take
Mm.
advantage of that by thinking about the approximation as essentially a function that goes from sensory inputs to probability distributions. And you could think about that, for example, as like a neural network where the weights are the parameters of that function. And so you're trying to find the weights such that the mapping from sensory inputs to probability distributions um, gets as close as possible to the target probability distribution, which is the posterior distribution given by Bayes' rule. But the
Mm.
whole point here is to kind of approximate Bayes' rule without actually implementing Bayes' rule, which is going to be computationally intractable. Essentially,
Mm.
it's like a feed-forward mapping that can be very fast, and that was that's um, the main reason to think about these kinds of approximations, because they solve this issue of how do you get really fast inference in
Mm.
neural circuits or other
Mm.
kinds of cellular systems.
Mm. So, and you mentioned that you're studying this, what's it called, stentor, or this
Mm-hmm.
other single cell animal. But if you want to study experimentally sort of the interaction between molecular memory and synapses, I guess you need a bigger animal, an animal with synapses, right? So, is this something which is like C. elegans, maybe, or
So
yeah?
there are different objectives here, or different intersecting objectives. So um, one objective is to try to isolate the processes contributing to molecular memory. And there
Mm-hmm.
you want to kind of get rid of synapses because that just complicates the picture.
Mm-hmm.
And that's why we started studying stentor. But then um, obviously we also want to understand how these two things interact with each other. And for that, you're right that we need organisms with synapses to study that. So that's why we've also started investigating planarian memory and this memory transfer effect, because
Mm-hmm.
we'd like to understand the nature of that memory transfer effect, which has never been understood before at a molecular level. And then ultimately, we would need to understand how that molecular memory interacts with synapses to produce behavior.
So do you actually do experiments in your lab, or?
Yes.
Oh,
Well, so right now the experiments with stentor are happening in my lab, and the experiments with planaria are happening in Michael Evans' lab
well,
at Tufts.
I'm so
Yeah.
also impressed that you're able to do both theory and experiment at this at the same time. It's
Well,
like,
almost everybody in my lab does experiments.
yeah,
I run an experimental lab.
yeah.
Yeah.
But
Yeah.
I mean,
Yeah.
it's I never done experiments, but just having to, I mean, dealing and learning about all the things. I mean, techniques and stuff in modeling. It's, and I guess it's an equal amount of details you have to learn to do experiments, right? So it's,
Yeah.
I
Well,
guess you
I mean, it
have bright
kind
students.
of yeah. I mean, you have to find the right people to do this
Yeah.
kind of stuff.
Huh.
Yeah.
Cool. Yeah. So, I mean, when I mentioned this, that I was going to have this podcast with you to to a colleague, I was out on this research school,
Hmm.
and then he said, well, I mean, he was working on working memory, so a different kind of memory, but he sort of he was open, but he said that maybe he has had a harder time to see how this molecular memory could play a role in more complicated memories, right? Other than sort of, I guess this single cell and this simpler, that's sort of like simpler aversive memories, and so it's not
Hmm.
like remembering your first day at school.
Yeah.
So what is your what's your comment to that? That is difficult to.
Well, we need to start somewhere, right? We need to
Mhm.
start with the simplest kinds of memories that capture something interesting, and then if we can understand that, then we can try to eventually scale up to more complex forms of memory.
Yeah.
Again, it's partly guided by this heuristic that evolution is conservative, and the mechanisms for storing complex information might not be that different from the mechanisms storing simple information. That's just a heuristic. It might be wrong. But I mean, if computer memory is any guide, right, that is how things work, right? We don't have fundamentally different forms of memory for storing, PhD theses and output, simple numbers, right?
Yeah.
Yeah.
Yeah. Exactly. Yeah. And we actually need also memory, separate memory in computers, right? So that's also, I mean, in the, if you think of the AI, right, at least, or the most commonly used neuronal unit is the ReLU unit, right? So which is doesn't really have a memory, or doesn't have anything. I mean, well, it only has this very simple sort of linear threshold kind of thing. So do you think this could be this interaction between molecular memory and synaptic memory that you envision, that could be something to useful for AI?
I'm not sure. I mean, people have different opinions about this, and I'm not sure which one is right. I think that it's clear from a biological perspective that cells, not just neurons, but all kinds of cells, are much more complicated than the units in neural networks. And people have even suggested that there's something like a neural network that's happening inside of a single cell.
Mm.
Um,
Yeah.
and that might actually be a better analogy to what we're calling neural networks than actual networks of neurons.
Yeah. We're going to have a
um,
podcast about that, upcoming podcast with what's called Benayota Bairazzi, who's like
yeah,
actually studying this like complicated
yeah,
dendrites, the kind of computation that a single neuron
yeah.
can do in the dendrites. Yeah.
Yeah, that's right. But then there are other people like I think Tony Zader would say that that level of detail doesn't really matter because there's some sort of universality argument, although I don't think this has ever been really spelled out rigorously, that um, you can get the same sorts of complexity just by adding more neurons that you get from just making a single neuron more complicated, right? Or it doesn't really matter. I mean, there's an interesting argument that's being unfolded currently within AI in deep learning research that suggests that the details of the function approximators don't matter all that much, because as long as the function approximators are expressive enough and you have enough training data and enough training time, enough compute, they all kind of produce similar answers, in the sense that if you look, for example, at the geometry of their underlying representations or just their outputs, they all sort of produce the same things, right? In the limit, right?
Yeah.
And so that's an argument that biological realism doesn't matter that much for AI.
Yeah. Of course, if you want to optimize energy consumption and stuff like that, then that might well change the story. Maybe that is some
Right,
kind
that's right,
of
and that might change the story in ways that we don't really understand yet. I mean, but another answer could be that yes, you need to constrain the amount of computation, amount of memory, and so on, but not necessarily in ways that look like neurons or biological cells. Right? There could be many different ways to do that, and maybe some ways might be even more efficient for our purposes than how nature does it. But it's certainly the case, right? It's inescapable to observe that nature has found a way to do really advanced computation much more cheaply than human engineers have, and so there's probably something worth learning there. I actually had, when they started the Kepler Institute, and they were purchasing lots of compute, and I suggested to them that if they were really serious about biological intelligence, they should limit compute to the wattage of a light bulb.
Yeah, that will. Yeah, that would be interesting. Yeah. Yeah.
Yeah.
So how did they respond? Well,
They didn't take me up on the suggestion, but I could have saved them a lot of money.
yeah, yeah, that's true. Okay, so we are approaching the end here, Sam. So anything, that you feel anything you would like to share?
No, thank you. This is an interesting discussion.
Yeah. So, like personally, I mean, memory has never sort of been my field, but I think it's in general, I think it's really cool that we are that you are exploring these ideas, which are a little bit sort of well, not in vogue, not the most modern. I think it's really important, and we should really be. I think we as a field should be really generous towards sort of like people exploring different ideas, because if we are honest, we don't really understand much of how the brain works, any of us. I would say so. I
Yeah.
think we should sort of explore many avenues. So,
Yeah.
yeah. Thanks a lot for coming to the podcast.
Yeah. Thank you.